Within the DNA, there are certain sections that code for protein. These are called genes.
Transcription is the first process that occurs in order for proteins to be made. Transcription occurs in 3 steps: initiation, elongation, and termination. In initiation, the RNA polymerase binds to the promoter. This is when the transcription of mRNA begins. The enhancer regions upstream of the promoter helps to speed up the process. The elongation process is when the RNA polymerase moves downstream and unwinds the DNA. As this is happening, the RNA transcript occurs in the 5' to 3' direction. The template strand of the DNA runs in the 3' to 5'. The complementary base pair rule occurs in this case too. However, instead of thymine being attached to the mRNA base, uracil is used instead. The transcription process ends in termination. Termination is where the RNA polymerase leaves its position in the DNA and the mRNA is completed. However, this is not the completed mRNA. It is called the pre-mRNA.
In order for the pre-mRNA to be completed, RNA modification must occur. Introns are removed by spliceosomes and then they join the exons together. A 5' cap is attached to the 5' end. This 5' cap consists of a modified guanine. A 3' poly-A tail consisting of a long chain of adenine is attached to the 3' end.
Translation is when protein synthesis occurs. The process of translation happens in 3 stages: initiation, elongation, and termination. The initiation starts when the mRNA attaches to the ribosome, which is made of 2 subunits, a large on top and a small on bottom. When mRNA attaches to the rRNA, the tRNA reads the mRNA code and translates it to an anticodon. The start codon AUG triggers the attachment of MET. Amino acid tRNA synthetase matches tRNA with the correct amino acid. The elongation process takes place as the tRNA goes to the A site, moves to the P site where the amino acid is added to the growing chain, and then it leaves through the E site when the amino acid is added. The elongation is able to occur because of peptidotransferase is able to join the single amino acids together. Finally, termination occurs when the tRNA reads a stop codon, which in our case is UAA in the A site. This triggers the release factor to bind to it, and the hydrolysis breaks the chain from tRNA. This process requires energy of 2 GDP molecules. Once that is done, the polypeptide is released through the exit tunnel and everything breaks apart.